We've all heard that the new mRNA vaccines use lipid nanoparticles as their drug delivery system. And since I knew next to nothing about that, I spent the last couple of hours researching it and figuring out what it is and how it works. And I think I've kind of figured it out to a very superficial extent at this point. If someone actually knows how this works and works in a lipid nanoparticle lab, please comment below and educate me. The last time I did anything remotely close to this was over 2 decades ago when I was in the lab transfecting plasmids into tumor cells with lipofectamine. Um, so the lipid nanoparticle basically tries to solve several problems with the mRNA vaccines. mRNA is an extremely fragile molecule, so it has to be protected from degradation. So you have to encapsulate it somehow, and then you have to get the particle into the cell. After you get it into the cell, you have to keep it from getting degraded by the cell and rather release the RNA into the cytoplasm so that it can be picked up by the ribosomes and translated into protein. So, the lipid nanoparticle is a way to try to kind of solve all those problems. So, I watched two videos on YouTube which were excellent, and they are basically talks at nanoparticle drug delivery system meetings, international meetings where scientists were giving their research data on how they were developing these lipid nanoparticles. So here's a slide from one of the lectures, and I'll link all these in the description. So what I found out is that these lipid nanoparticles actually simulate LDLs and HDLs. And so apolipoproteins will actually bind into them, and then LDL receptors or HDL receptors will cause receptor-mediated endocytosis. And then once you get endocytosis, it becomes an endosome. And then pH changes in the endosome are supposed to make the lipid nanoparticle fuse with the phospholipid bilayer and then release it into the cytoplasm. Um, so the major problem here, evidently, is that after it gets into the cell as an endosome, the vast majority of these things go into the lysosome and get degraded and are just totally eliminated. And a very small percentage escape the endosome and you get translation of the protein. So this is one of the major things that they're trying to fix right now, and evidently this is something that has not been solved very well, and still you get the vast majority of the vaccine will end up being degraded immediately by the lysosome and not creating a functional response. So, okay, so in this lecture, they described how these things are created. And what it is you take the mRNA that you want to encapsulate in an acidic buffer, and you place it in one well, and then you take the four components in ethanol and put it in another well. And so these four components, after looking into it, are these. One is going to be a cationic lipid, and that binds the RNA. And then cholesterol and this cationic lipid bind the RNA and stabilize it and becomes the central core of the nanoparticle. Then there is a phospholipid that forms the inner shell. And so these phospholipids are kind of like the phospholipid bilayer that we have that surrounds our cells. And then there's a pegylated lipid that forms the outer shell. And evidently, the amount of pegylated lipid you put in here, the concentration, determines the size of this nanoparticle. So you can adjust the size of the nanoparticle with this. They run it through, they fuse in a well, and then you dialyze it to purify it. I looked at the components of the Pfizer vaccine and figured out that basically this long thing right here, this is the cationic lipid, this is the pegylated shell, this is the phospholipid that forms the inner shell, and then cholesterol. So those are the components of the lipid nanoparticle in the Pfizer vaccine. And so, yeah, after it gets fused, a small percentage then gets endosomal escape, then it becomes translated here, and then here's the translated protein. The protein then gets degraded by a proteasome and then gets presented on the MHC1 cell. Excuse me, the MHC1 complex, and the peptide gets presented on MHC1. And then if this cell gets degraded and then taken up by an antigen presenting cell, it will present on an MHC2. And so that's how you get the immune response from these mRNA vaccines, which is interesting. Then I tried to find out, is there any lipid nanoparticle that's been approved for human use? And there's exactly one, and it's this one. The trade name is Onpatro, and the generic name is Patisiran. Evidently, this is not an mRNA drug. This is an siRNA drug, small interfering RNA, and it's to treat this really rare disease called transthyretin-mediated amyloidosis. And they had a phase 3 clinical trial with 200 and some patients. It's a very rare disease of about 50,000 people worldwide. But the important thing here is they did this test. They followed 218 patients for up to 4 years, and they did not find any serious side effects. So that's very encouraging that at least we know that there were not serious side effects in 218 patients. Now, this is not a huge number of people, so there may be some extremely rare side effects that are not picked up in a study of this size, but they did follow for 4 years, so that's fairly long-term follow-up. This thing got approval in late 2018, right here. And then in terms of clearance, these things are not cleared by the kidney, so they're too big to go through the renal filtration threshold, and so they have to be broken down intracellularly and eliminated intracellularly. And then the components have to be broken down, and then they can be excreted. So the way I got into this is there was this letter that I kind of responded to in another video, and he talks about here, talking about the pegylated lipid nanoparticles, and makes a bunch of claims about how they could be dangerous. And I went through every single one of these, and none of them talked about the toxicity of lipid nanoparticles, except for this last one, which is right here, which is a great paper, actually. And in the end, they do mention some toxicity concerns right here. So amongst the most problematic are the potential toxicity of lipid nanoparticle components, including the cationic lipids, phospholipids, or combination thereof. So we don't know the toxicity. It's kind of new, potential. We know that lipofectamine was very toxic when people tried to do that in vivo. And so that's why they moved to these lipid nanoparticles, which are thought to be much less toxic. But any drug is going to be toxic to some extent. And it's unclear. We know that from this drug, in 218 patients, there were not significant toxicity, but we don't know what the exact toxicity profile of these nanoparticles are. And then PEG can elicit an immune response in and of itself with repeated doses. So with a vaccine that's dosed once or twice, it should not be a major factor. But the interesting thing is these drug delivery systems are, they are definitely happening. And you can see there's a long list of drugs that are nanoparticle and liposomal drug delivery systems that are currently approved. And there's hundreds more in clinical trials now. And, when I was in the lab, a long time ago, we were talking a lot about gene therapy and targeted therapy, and one of the major problems was a drug delivery system or a gene delivery system, and it looks like we're very close to a good gene delivery system, it's very exciting actually that this mRNA delivery system seems to be working. And this is a drug delivery platform that probably is going to become very common. So I think it's important that we kind of understand how these things work. And I think the toxicity information will become more clear as more and more of these type of drugs get approved. Um, the major issue in this case is that a lot of these nanoparticle drug delivery systems are for cancers. And in cancers, we tolerate a lot of toxicity. But in a vaccine, you just can't tolerate high toxicity because you're giving it to healthy people. And so it needs to be extremely safe. And so this, I think, vaccine rollout in healthy people is going to be a real test of the toxicity of this drug delivery system. Um, I'll link the other video about this letter response, and thanks for watching.